Hello.
Hello. I see we have our keynote lecture. So we can start. Hello, everyone. It is really nice to see so many people joining us today to our second keynote lecture, The Neural Control of Immunity, Mechanisms and Implications for Treating Patients. Just 1 hour ago, we had some news that due to urgent reasons, Professor Kevin Tracey won't be able to join us. I'm so sorry for that. But we're really happy that his colleague is taking a lead and will be giving his lecture today. So I'm so honored to present Dr. Sangeeta Shivan. She's a professor at the Feinstein Institute for Medical Research and co-author of more than 80 peer-reviewed scientific research papers and reviews. She is an inventor of multiple patents and patent applications and has traveled nationally and internationally to deliver talks at scientific meetings. While studies led by Dr. Kevin Tracey discovered a vagus nerve mechanism turned the inflammatory reflex that regulates the immune system, Dr. Shivan's studies have been instrumental in mapping the afferent and afferent vagus nerve pathways in the inflammatory reflex. So Dr. Shivan's research more focuses on studying the neural circuits that regulate immune responses. So thank you, lecturer, for joining our conference. And without further ado, please welcome Dr. Sangeeta Shivan.
Yeah, hi. Can you see me and hear me?
Yes.
Yeah, okay. Sorry for being a couple of minutes late, and hello, everyone. I was just told an hour ago to give like. I'm a professor at the Feinstein Institute for Medical Research. I work very closely with Dr. Kevin Tracey. We are spearheading multiple projects together in the lab, and the work that I will show you has been done in collaboration with Dr. Tracey. So let me start sharing my slides here. Mm-hmm. Oh. Alicia, do you see my slides?
No, we cannot see your slides. You should press plus and then your presentation.
Do you see those now?
No.
Okay. Not yet, right? I'm sorry for this. So I'm sharing my PowerPoint. It gives me permission to access screen is not granted.
You can try sharing your screen and showing us your presentation.
Do you see anything right now?
No, we cannot see it.
Okay, let me log off and log in again. Maybe there is a problem here. I'm so sorry for everyone for this. Hello, Elijah.
Hello.
Okay, so you can hear me now. Let me see if I can share slides. Do you see those now?
No, unfortunately, we cannot see it. We think maybe you should give permission to your browser to show.
Yeah, I did that. I just did that because that was a problem actually. Elijah, I have done everything. I'm not sure. Is there anyone on your side who can help me?
We believe you could send us your presentation and we could upload it.
And then you will hand, do I get a control of presentation over there? Or how is
Yes.
it?
Yes.
Okay. Yeah, let me do that. Give me 1 second.
Okay, thank you.
Mm-hmm. The file is too big to send. Okay, give me can everyone please give me 5 minutes? Let me figure out.
Yes, of course.
Hello.
Hi.
I have emailed a presentation. Did you receive it?
We will check in a few seconds. Yes, I believe we got it, and our member is uploading it now, so everything will be okay.
Great.
Hello, I just wanted to check, is it okay if I will share my screen so you could talk and I will change the slides for you, or it is better if I upload the file? Because if I upload the file, I'm not sure if the format is gonna be okay and if the videos are gonna be okay. I'm sorry we not hear you, we cannot hear you.
Yeah. I mean, that's okay with me. It's just that it will be better if I have the control because there is a lot of animation in the presentation. Why? Is it possible to do that or no?
I will try to upload maybe, and then we will see if the animations are working, okay?
Okay, let's see. So with all these delays, I mean, how much time do we have for the presentation now?
As this is the last part of our conference, we have all the time in the world, as long as
Okay. Yeah, okay. No, I was just trying to figure out whether I should rush through the slides or just not talk about some slides. Yeah.
I'm sorry, but I cannot upload the presentation because the file is too big somehow. So I will try to share my screen and maybe it's gonna be okay. Just tell me if it's okay.
Yeah, okay, let's try that then. I mean, if I'm not able to share my screen, that's the. So, Joanna, do you know if I can share my screen? I mean, how can I enable that?
So I'm not sure if you see, but there is like this button here.
Yeah, blue
So
button.
maybe, yeah, this one on the right, the last one. So I could give you the rights to do it. So maybe you can try. Maybe it will be okay for you too.
Okay, let's try one last time. If it is not, we will go by your thing. Okay.
I believe you should have a right to do it. Do you see that blue button?
Yeah. So I'm trying to share my presentation. The problem is I don't see the presentation when I go to that window. That's the problem, I think, here. Let me see if I can bring it here. That is what is happening. I don't see it in your screen at all.
Yeah, I stopped sharing my screen now, so maybe you could try to share your screen by pressing the same button, which is like the one on the right.
Do you see my screen now?
No, we don't see it.
I don't know. I'm not able to select the PowerPoint file in when I do the share screen. That is what is happening.
But did you press that button I show you, the one on the right?
Yes. So I press the button. It gives me an option to choose what to share, entire screen, window, or Chrome tab. I'm selecting the window that has Microsoft PowerPoint, and I'm sharing that, but it is not sharing.
Maybe you should try sharing the screen or desktop. Maybe that's the problem.
Okay, so I'm sharing the entire screen now. Do you see my screen?
No, I'm sorry, we don't.
Yeah, I know, it's not sharing the screen. Okay, let's go with that. Come on, because people are waiting for a long time. Yeah.
Okay, so if you need me to move something, you can just tell me and just see if you see the presentation in the full screen.
Yes, I see the presentation now. Okay. So hello everyone again. I'm extremely apologize for this delay in the presentation and all the technical difficulties. So to introduce myself, I'm Sangeeta Chavan. I'm a professor at the Feinstein Institute for Medical Research in the Institute of Bioelectronic Medicine Center. I have been involved in, my interest is understanding the neural control of immunity. And I have been working in collaboration with Dr. Tracey for more than a decade now. What I will be showing you for the next few minutes is our collaborative work for the past many years that have mapped the inflammatory reflex neurocells and have gone to the clinical applications. So can we go to the next slide, please? So we know that cytokines, inflammatory cytokines such as TNF are both necessary as well as detrimental to the physiology. These cytokines lead to the cytokine storm as we are seeing right now in the current pandemic situation. So Dr. Tracey's previous studies have shown that if you block these cytokines, you can have beneficial effects. Yeah, please go to the next slide. So in this study, which was published in Nature many years ago, what Dr. Tracey had done was, if you press the click, Jonah, yeah, keep on going with that thing, yeah. So when you give infection to the baboons, they make increased levels of cytokines. And block these cytokines using cytokine-specific antibodies, in this case, anti-TNF antibodies, that protects these animals from going into the lethal shock and death. So based on that, a cytokine theory of disease was proposed. Next slide, please. Which says that similar to germ theory, that tells us that all the germs are not bad. Similar to that, all the cytokines are not bad. Cytokines are required for health, such as they carry or they mediate many essential cell functions, such as cell recruitment, angiogenesis, etc. But when these cytokines are produced in excess, they lead to a different pathological conditions. Fever, anorexia, fatigue, syndrome, and so on and so forth. But one in which disease, it has been possible to develop anti-cytokine therapies, such as anti-cytokine antibodies. Can you press click, please? Yeah. So these anti-cytokine therapies have been in clinic for many years, and they have been used for many different diseases. And they comprise a huge market into the pharmaceutical world, more than $50 billion in the current scenario. And that is around 5% of the total world pharmaceutical market. So we and others have extensively studied these cytokines in different disease conditions. Next slide, please. And diseases ranging from, next slide, please. Ranging from ischemia, no, previous slide. Many diseases such as ischemia, reperfusion injury, hypertension, cancer are mediated by cytokines. So in our earlier days, we were interested in identifying small molecule inhibitors that will inhibit the cytokine storm and have protective effects. Next slide, please. One of our target molecule was CNI 1493. This is an arginine analog or homolog, and that we had developed in the lab and studied in many inflammatory disease preclinical models. Next slide, please. Click. So we showed that when you give the CNI 1493 into micromolar amounts to animals with inflammatory diseases, this protects the animals from these inflammatory diseases. This CNI 1493 has been extensively studied in our lab and has published here. But in this case, we were giving the CNI 1493 systemically, like we were giving intraperitoneally. So as a next step, we were interested in finding out whether this CNI 1493 can also inhibit inflammation in case of stroke. And to our surprise, we found that when we give CNI 1493 into the brain, it attenuates the inflammation in the brain as well as it attenuates the inflammation in the periphery. Can you please go to the next slide? And the interesting point was the CNI 1493, when it was given in the brain, we needed 1,000-fold less quantity of CNI 1493. That was suppressing inflammation in the brain as well as in the periphery. And this was quite puzzling because at this point, we did not know what is mediating the effect of CNI 1493. Around this time, we became aware of the work by Linda Watkins. We showed that vagus nerve may be playing a role in mediating signals from the brain to the periphery and from the periphery to the brain. So to check that, we cut the vagus nerve in these animals and gave CNI 1493 into the brain. And we found that when the vagus nerve signals are cut or the CNI 1493 no longer suppressed inflammation in the periphery, indicating that the vagus nerve was mediating the signals from the brain to the periphery and suppressing inflammation in the periphery. Next slide, please. So that was quite interesting. What we see here in the middle is the vagus nerve, which looks like a jellyfish, right? This is a picture that was drawn by Vesalis in has around 100,000 fibers. 80% of those fibers are sensory and 20% are the motor. So 80% of the fibers in the vagus nerve carry information from the periphery to the brain, and 20% carry information from the brain to the periphery. So many studies have been carried out to figure out the role of vagus nerve in modulating function of different organ systems. Next slide, please. And they have shown that vagus nerve plays a role in controlling heart rate, blood pressure, respiratory rate, insulin secretion, gastrointestinal function, etc. But what is not on this list is the immune system. It was not known earlier whether vagus nerve can play a role in modulating responses in the immune system. neural reflexes. Next slide, please. So Dr. Charles Sherrington, next slide, please. Yeah. Dr. Charles Sherrington taught us that neural reflexes are simple neural reflex circuits where efferent signals are transmitted to the cell body or to the central nervous system, where they are processed and then the efferent signals are originated. Once we understand how this neural reflex circuit works, we can understand how the nervous system works. That is what Dr. Sherrington proposed. But it was not that easy, right? Because the nervous system is an interlinked system. So if based on this, we hypothesize that the nervous system using neural reflex circuit is also controlling the inflammation. And that's where inflammatory reflex came into picture. Next slide, please. Is that the peripheral signals, inflammatory signals, are transmitted by the vagus nerve to the central nervous system, where it initiates number of pathological events such as fever, anorexia, etc. And that leads to an efferent signal which controls the peripheral inflammatory responses. So understanding this was very important because you can now directly activate or stimulate the vagus nerve using electricity and suppress peripheral inflammation. And that is what the data I will be showing you now. Next slide, please. So to address the role of the vagus nerve, we used an endotoxemia animal model. This is a picture, or this is a data from endotoxemic rats, where our graduate student Ludmila, when she gave TNF, as shown in the empty bar over here, but when she cut the vagus nerve in these animals, they made more TNF, as shown in the bar next to that. But when you stimulate the vagus nerve electrically in these animals, the level of TNF is significantly decreased, indicating that vagus nerve stimulation is suppressing inflammation in these animals. In addition to suppressing inflammation, these animals are also protected from going into shock, as shown in the right-hand side graph, where when the endotoxin is given to the animals go into shock quicker. But when the vagus nerve is stimulated, as shown with the squares over here, the animals are protected from going into the shock. So what we had discovered here, accidentally rather, was the next slide, please, Joanna. So inflammatory reflex is a neural reflex circuit in which the signals transmitted in the vagus nerve are processed in the brain and efferent signals are generated. These efferent signals then control inflammatory responses in the periphery. We then carried out extensive studies to map the afferent or the sensory signals in the vagus nerve as well as motor signals in the vagus nerve in this inflammatory neural pathway. Next slide, please. So to map the efferent arc of the reflex circuit, we carried out extensive studies. Which, when you stimulate the vagus nerve signals are inhibiting cytokine production in the spleen. And that was very important because a spleen has only one nerve going in and one artery going in. So using the spleen and endotoxin as a model, we could map this neural reflex circuit. Next slide, please. We now know that when you give endotoxin to the animals, more than 95% of the TNF is made in the spleen. What we see here, the red dots are the splenic macrophages, both red pulp macrophages and marginal zone macrophages that make TNF when the animal is given endotoxin. But when you subject these animals to vagus nerve stimulation, as shown in the bottom panel over here, is significant by the splenic macrophages. We next identified the receptor that is expressed on the splenic macrophages and that is required for the effect of vagus nerve stimulation. Next slide, please. Yeah. Here, when the wild type animals are subjected to vagus nerve stimulation, there is significant suppression of TNF production, as shown in the dark bar over here. But when you knock out a nicotinic acetylcholine receptor alpha 7 subunit, this effect is abolished, as shown in the shaded bar over here. So this indicates that the nicotinic acetylcholine alpha 7 receptor is absolutely essential for mediating the inflammatory effects of vagus nerve stimulation. And that was interesting to know because the splenic nerve is adrenergic nerve and it is not cholinergic nerve. This alpha 7 receptor is acetylcholine. So what was making acetylcholine into the spleen? Our studies identified a unique T cell subset in the spleen that was making acetylcholine and was required for this pathway. Next slide, please. What we see here are the green cells or green T cells. These T cells are green because they are making GFP, they are expressing green fluorescence protein under the control of CHAT or choline acetyltransferase enzyme. So this is a long way of saying that these T cells are green because they are making acetylcholine. Choline acetyltransferase is an essential enzyme in acetylcholine biosynthesis. So we found that these acetylcholine producing cells, which is shown in red over here. And these acetylcholine producing T cells are absolutely required for the anti-inflammatory effects of the vagus nerve circuit. Next slide, please. So when these T cells are present in the animal, when you knock these T cells from the animals, as shown over here, you lose the effect of vagus nerve stimulation. But if you reconstitute the animals with these T cells, acetylcholine producing T cells, you restore the effect of vagus nerve stimulation, indicating that these acetylcholine producing T cells are absolutely required for this pathway. Then Peter Olofsson in the lab, he further characterized these T cells and found that these T cells are a unique T cell subset. He used the ImGen dataset available these T cells with another 198 known immune cell subset and found that these T cells are the only T cells which are making high levels of choline acetyltransferase. In other words, they are making high levels of acetylcholine. So what we now know is that, can you go to the next slide, please? That signals in the vagus nerve terminate onto the spleen, onto these T cells that make acetylcholine. So the acetylcholine, which is made by these T cells, in turn binds to the macrophages and monocytes via the alpha 7 receptor and suppresses cytokine production in this pathway. So what we have identified, or what I have shown you so far, is the efferent of the matrix. The next question was, what is the brain origin in the brainstem that leads to activation of these signals? So the cholinergic fibers in the vagus nerve are known to originate into two distinct brainstem nuclei, the dorsal motor nucleus of the vagus and nucleus ambiguus. It has been known that the fibers, the efferent fibers, which originate into the dorsal motor nucleus of the vagus, innervate a number of visceral organs. So we asked a question whether the fibers which are originating into dorsal motor nucleus of the vagus are also controlling inflammatory responses in different organs or not. So to address that, we utilized a specific transgenic animal Next slide, please. Channel rhodopsin onto the cholinergic neurons in the dorsal motor nuclei. What I'm showing over here is the dorsal motor nucleus of this particular transgenic mouse, where the cholinergic nuclei, which are shown in the red over here, are expressing the channel rhodopsin shown in the green over here. We utilized this mouse and selectively targeted the dorsal motor nuclei under stereotaxic guidance. Next slide, please. And then we stimulated these cholinergic neurons in the dorsal motor nuclei using optogenetic modality. What we see here is that when the cholinergic nuclei in the dorsal motor nucleus are stimulated, it suppresses cytokine production in response to endotoxin challenge in these ants. So what we observed was, and shown for the first time, the origin of these signals into the brainstem nuclei of the mouse. Next, we wanted to identify whether the signals which are originating into the brainstem are directly going to the spleen or they are going via the celiac ganglion. Next slide, please. It is known that the vagus nerve does not directly innervate the spleen. The vagus nerve fibers that terminate into the celiac superior mesenteric ganglionic complex, which is present in the peritoneal cavity. And the splenic nerve originates in this celiac superior mesenteric ganglionic complex. So it was very intriguing and surprising how the signals which are originating in the vagus nerve or in the splenic nerve. So to address that, we map the signals which are originating into the dorsal motor nuclei using tracer viruses. Next slide, please. We use viral tracers where adeno-associated virus, AAV, was injected into the dorsal motor nucleus. This AAV is an anterograde virus. That means it travels from the cell bodies of the nuclei of the neurons to the axons of the neurons. And this virus expresses the yellow fluorescence protein, which is shown in green over here. At the same time, in same animals, we injected HSV or the herpes simplex virus into the spleen parenchyma of these animals. This HSV expresses MJ, which is shown. And then we harvested celiac ganglion from this mouse and then carried out immunohistochemistry. What we see here is that the green vagus nerve fibers are forming, are rather terminating in very close proximity to the splenic nerve cell bodies, indicating that the signals which are originating into the vagus nerve or into the dorsal motor nucleus of the vagus are being transmitted by the vagus nerve fibers and are terminating onto the splenic, into the celiac ganglion, onto the splenic nerve. And then they are transmitted by the splenic nerve to the spleen. To confirm that, next we recorded on the splenic nerve. So we put an electrode onto the splenic nerve of the mouse and then recorded in real time when we were stimulating the dorsal motor nucleus. Next. So in these animals, we stimulated the dorsal motor nucleus using light and then recorded onto the splenic nerve in real time. So the bars in the egg show that in the legitimate control that do not express channel rhodopsin, if you shine light as shown in the blue boxes over here, there is no difference in the splenic nerve activity. But in chat channel rhodopsin animals that express the channel rhodopsin onto cholinergic neurons, when you stimulate these cholinergic neurons using light, there is increased splenic nerve activity. Further confirming that the signals which are originating into the cholinergic neurons in the dorsal motor nucleus of the vagus are being transmitted to the celiac ganglion and in turn are activating the splenic nerve, leading to splenic nerve over here. Next slide, please. So these studies have extensively mapped the motor arc of the inflammatory reflex circuit, where we now know that signals which are originating into the dorsal motor nucleus or the vagus are transmitted to the celiac ganglion and from the celiac ganglion to the periphery and thus suppressing inflammation. So we have utilized this concept in bioelectronic medicine. Next slide, please. Where now we are asking a question that instead of using anti-cytokine therapies such as anti-cytokine antibodies, can we use vagus nerve stimulation directly to suppress the cytokine responses in different inflammatory disease conditions? To address that, next slide, please. Such as rheumatoid arthritis and inflammatory bowel disease. So to address that, we carried out a clinical study in collaboration with Setpoint Medical. This study was led by Dr. Frida Cookman and Dr. Paul Petertak and Dr. Ralph Zetnik. Here, rheumatoid patients were implanted with vagus nerve stimulators into the cervical region, as shown in the picture on the right-hand side. The electrodes were implanted onto the cervical vagus nerve, and then the pulse generated was placed into the chest cavity. And these subjects were then followed for more than 2 years now. So in the initial study, we first wanted to find out whether there is a placebo effect. So rule of epilepsy patients. Next study slide, please. So here, epilepsy patients who were undergoing this vagus nerve stimulation as a therapy were subjected to vagus nerve stimulation, and then we analyze the inflammatory responses in their whole blood. As we see here, after a single vagus nerve stimulation, there was a significant decrease in inflammatory cytokine production in these epilepsy patients. And this was of interest because these patients were anesthetized. So as we know that these patients do not know the difference between the placebo and the real stimulation, right? Because they are anesthetized over here. So this was the first demonstration that electrical stimulation of the vagus nerve leads to suppression of the cytokine production in human subjects. Rheumatoid arthritis patients. Next slide, please. Where we enrolled 17 patients in this particular clinical trial. These 17 patients were implanted with vagus nerve stimulator on day 0, as shown over here. They were subjected to single electrical vagus nerve stimulation, and then the vagus nerve stimulator was switched off. And after 7 days, the stimulator was turned on, and then it was kept on for 42 days, and again it was switched off. So the blue bars over here are showing the period when the vagus nerve stimulator was switched off. So as we see here, that when the vagus nerve stimulation was switched on in this patient, the disease activity was significantly decreased over time. When the stim was turned off from day 42 to day 56, the disease activity increased significantly in these patients. We also tracked the TNF levels in these patients in the whole blood over time during this study period, which is shown in red over here. So again, the TNF levels tracked significantly, or rather correlated significantly with the disease activity as shown in the graph over here. So we showed that the vagus nerve stimulation in this rheumatoid arthritis patient inhibited both cytokine production as well as attenuated the disease severity in this patient population. Next slide, please. So this is Dr. Tracey on the left-hand side here. He made the first patient who was enrolled in this clinical study. He is a 45-year-old male, and he had 8 years of rheumatoid arthritis disease history. And with this particular treatment, now he is in clinical remission completely. And we have been following these patients for more than 2 years now, and these patients are in clinical remission still. Next slide, please. This is another patient who was covered by New York Times. And again, she was suffering from the rheumatoid arthritis patients for many, many years and had a lot of problems because of the disease. Could not even pick up a pencil, could not even needed help with clothing, etc. But after this vagus stimulation trial, and she is able to ride a bicycle to the Dutch coast and nearly 20 miles down from home. Therapy has been helping many such patients currently. So the next slide, please. So after mapping this efferent pathway in the inflammatory reflex circuit, so the next question was how the afferent or the sensory signals are transmitted in this pathway. If you go to the next slide, the vagus nerve act a major high in the body. It's like it is listening to these inflammatory conversations between the periphery and the brain. And it is carrying out all these significant signal transmission between the periphery and the brain. So to understand how these signals are transmitted via the vagus nerve to the brain, we developed a system in the lab, a preclinical system, where, next slide please, onto the mouse vagus nerve in real time in response to cytokine challenges. What we see here on the right hand side is a vagus nerve in a mouse, which is less than 100 micron in diameter, which is shown by a white arrow over here. So we very carefully isolated the vagus nerve in the cervical region of a mouse and placed it on the electrode and challenged the animal with cytokines. If you go to the next slide, please. And recorded the signals in these animals. So when the animals are challenged with vehicle, in this case saline, there is no change in the vagus nerve activity as shown by the arrow over here. But if you challenge these animals with either TNF or interleukin 1, next please. You see significant increase in activity. Next please. With both TNF and interleukin 1. The important thing to note here is that that the signals which are generated in the vagus nerve in response to either TNF or the interleukin 1 are distinct, both in the temporal responses and in the amplitude. So to next slide. So to address whether these signals are cytokine receptor dependent or not, we utilized receptor knockout animals. As shown in the top panel over here, both TNF receptor and interleukin 1 receptors are expressed on the sensory vagus neurons from the nodus ganglia. And when you knock out this TNF receptor and interleukin 1 receptor in animals, reduced as shown in the bottom panels over here, indicating that the activation of the vagus nerve in response to these different cytokines is cytokine receptor dependent. Next slide, please. So to understand this, we carried out recordings in response to different cytokines such as IL-1 and TNF in real time in these animals. And then we developed algorithms to isolate and decode this neural activity which was recorded on the surface of the vagus nerve. Using these Bayes algorithms, we identified different neural subtypes or the subgroups that are firing in unison in response to cytokines. So what we see here on the right-hand side graph is one group of neurons, which is shown in yellow, that does respond. In contrast, another group of neuron, which is shown in orange over here, that is called as a CAP cluster 2 or the compound action potential cluster 2. It responds to interleukin 1 challenge, but does not respond to TNF challenge. Whereas the third neuronal group, which is shown in blue over here, responds only to TNF and not to interleukin 1, indicating that the vagus nerve is transmitting signals in a cytokine-specific manner. And this is really important to develop and decode these signals so as to develop the diagnostic and closed-loop circuits in the future. So next slide, please. So this was using these principles. We are carrying out studies in our medicine, understanding the molecular targets which are required or which are mediating different inflammatory diseases, and then identifying the neural circuits which are controlling these molecular targets. And at the same time, we are using technology to develop devices to modulate these neural circuits so as we can then control these molecular targets in different disease conditions. And we are recruiting. So please feel free to reach out to us if you are interested in joining our exciting group. Next slide, please. So this is our typical lab meeting, and this is the lab which is having and discussing our scientific projects over here. And thank you. I'll be happy to take any questions.
Thank you for the wonderful lecture. Maybe we have some questions from the audience. Okay, so thank you for the fascinating lecture. It was really interesting. And I think I'm speaking on behalf of everyone who will listen to you.
Thank you.
So, second day of International Health Sciences Conference 2021 is over, and conference is officially ended. I hope these 2 days showed you that science, one way or another, can thrive even during the pandemic. And next year, I hope we will be able to meet in person as we used to. From all the organizing committee, I say thank you, everyone.